Hello everyone, my name is Kenny Coster and today I'll be presenting one of the most powerful and controversial technologies in modern biology, CRISPR gene editing. Over the next several minutes, I'm going to walk through what CRISPR actually is, how it works, and what makes it so revolutionary, and why scientists, ethicists, and governments around the world are having urgent discussions about its use. We'll look at the scientific breakthroughs, medical possibilities, and also the ethical concerns, like genetic inequality, designer babies, and how CRISPR could reshape the future generations. My goal is to make this complex topic accessible and to show why CRISPR is both incredibly promising and potentially risky if misused. Gene editing means making intentional changes to genomic DNA, insertions, deletions, or replacements. Earlier targeted nucleases such as zinc-fingered nucleases and tannins permitted targeted cuts, but CRISPR introduced a much simpler RNA-guided approach that de demonerized genome editing across labs. CRISPR arrays were first observed in bacteria in the late 1980s, but the demonstration of the programmable Cas9 mediated DNA cleavage in vitro was established by Yannick et al. In 2012, which set the stage for broad applications. So first we got to talk about is what is CRISPR? CRISPR stands for clustered regularly interspaced short palindromic repeats, and originally comes from a natural immune system found in bacteria. In 2012, Jonathan Dundra and Emmanuel Carpentier discovered that the system could be re-engineered into precise gene editing tool. The easiest way to think about CRISPR is like a programmable pair of molecular scissors. Scientists can design a short piece of RNA that guides the CRISPR protein, usually Cas9, to a specific DNA sequence. Cas9 then cuts the DNA at that location. This ability to search and cut DNA with such accuracy has completely transformed genetic engineering. Before CRISPR, gene editing was slow, expensive, and often inaccurate. Now it's fast, affordable, and programmable with incredible precision. So let's break down how CRISPR works in a simple way. Step one, a scientist designs a guide RNA that matches a DNA sequence they want to target. Step two, the guide RNA and Cas9 enzyme pair together and the guide RNA leads Cas9 directly to that DNA segment. Step three, Cas9 makes a cut in the DNA. Once the DNA is cut, the cell tries to repair the break. And this is where the gene editing happens. There are two main repair pathways. The first is called the non-homologous end joining. Often makes small errors, and these errors can knock out a gene and turn it off. The second, called homology-directed repair, can be used to insert new DNA or fix a mutation. This ability to precisely cut and rewrite DNA is why CRISPR is such a powerful technology. CRISPR's medical potential is absolutely enormous. It offers the possibility of treating or even curing many genetic diseases at their source by correcting the underlying mutation. Some examples include sickle cell disease, cystic fibrosis, muscular dystrophy, and certain forms of inherited blindness. In 2023, the FDA approved the first CRISPR-based therapy for sickle cell disease, which is a huge milestone for medicine. Clinical trials have also shown promise in using CRISPR to enhance immune cells to fight cancer, and even to reduce viral infections like HIV. Most of this work using somatic editing, Most of this work uses somatic editing, meaning the changes happen only in the treated person's cells and are not passed on to future generations. Somatic editing is widely considered acceptable is considered ethically acceptable as long as it's safe and controlled. Now things get more ethically complicated when we talk about editing human embryos. Editing embryos means the changes affect the germline, which means they're passed down to future generations. In 2018, a Chinese scientist named He Jiankui shocked the world by secretly editing embryos to create the first genetically edited babies. He claimed the edits would make the children resistant to HIV. The procedure was unsafe, lacked proper oversight, and the full consequences of the edits are unknown. Many countries responded by triggering or tightening laws and calling for global guidelines. Gene editing in embryos raises questions not just about safety, but about human identity, consent, and whether we have the right to make permanent changes to future generations. Oh, we're going to talk about some ethical concerns now. When scientists and ethicists talk about the dangers of CRISPR, one of the biggest concerns is safety. Even though CRISPR is precise, it can still make mistakes called off-target mutations. These are unintended cuts that the genome might cause harmful effects, like cancer or developmental problems. Another risk is mosaicism, means not all the cells in the embryo get edited the same way. Some cells might carry the edit, while others do not, creating unpredictable outcomes. The truth is, we still don't fully understand the long-term consequences of altering genes, especially in embryos. Human biology is complex, and traits often involve many genes working together. Changing one gene could have a ripple effect we don't yet understand. Another major ethical issue is genetic equity. CRISPR treatments are expensive, and if gene editing becomes part of medicine, there's a real risk that the only wealthy people will have access to it. Imagine a society where some people can afford to correct genetic conditions or enhance certain abilities, while others cannot. This could create a new form of an inequality, one based on biology itself. Equity also relates to global justice. Many countries have fewer resources to regulate or access advanced technologies. Without careful governance, CRISPR could widen the gap between nations and between socioeconomic groups. So one of the biggest questions is, how do we ensure the benefits of CRISPR are accessible to everyone? The phrase designer babies is often used when people talk about gene editing. This refers to using CRISPR to select or enhance traits that are not medical necessities, like height, intelligence, athletic ability, or appearance. Scientifically, these traits are extremely complex and depend on hundreds or even thousands of genes, environment, or even random chance. We are nowhere near being able to safely edit them. But the idea of designer babies raises important questions. Would this create unrealistic expectations for children? Could it reduce human diversity? Would parents feel pressure to edit their kids to stay competitive? And who decides what counts as a desirable or acceptable trait? These concerns highlight why most scientific organizations oppose any form of enhancement editing. Another issue is the impact of gene editing on human diversity. Our genetic differences are part of what makes us adaptable as a species. Eliminating certain genes could reduce that diversity and potentially remove traits that might someday be useful. There is also an emotional and cultural dimension. Some traits or conditions, such as deafness or neurodiversity, are part of a people's identity and communities. Editing them out could unintentionally send a message that certain lives are less valuable. So the question is, how do we balance the desire to reduce suffering with the need to respect the full range of human experiences and identities? Because of CRISPR's power, many organizations argue that global rules are necessary. The World Health Organization, or the WHO, and the U.S. National Academies have recommended strong oversight for all human gene editing research. They emphasize that somatic editing the non-reproductive cells, can move forward with caution, but germline editing should not be allowed for now. They also call for transparency, international cooperation, and public involvement in decision making. This isn't just a scientific issue, this is a societal one. The future of gene editing will affect everyone, so everyone deserves a voice in how this technology is used. Let's revisit the He Jiankui case because it highlights why strong governance is important and needed. He edited the CCR5 gene in embryos to supposedly make them resistant to HIV, but the procedure was rushed, unsafe, and unethical. He bypassed regulations, misled parents, and didn't fully understand the consequences of altering this gene. The children born from this experiment may face unknown health risks for the rest of their lives. The global scientific community responded with outrage, calling it irresponsible and reckless. This scandal shows what can happen when powerful technologies are used without oversight. It also pushed the world to create clear guidelines to prevent something like this from ever happening again. With all of these concerns in mind, how do we move forward responsibly? The truth is, CRISPR has extraordinary potential. It could eliminate devastating genetic diseases and improve the quality of life for millions of people. But with that power comes tremendous responsibility. We have to balance scientific innovation with ethical caution. That means addressing safety concerns, ensuring equitable access, protecting future generations, and making sure gene editing is used to promote health and well-being, not to divide society or reinforce discrimination. CRISPR is a tool, whether it becomes a force for good or a source of harm, depends on the decisions we make today. So to quickly summarize some things up, CRISPR is absolutely groundbreaking technology that allows precise editing of DNA. It has immense medical potential and could transform treatment for genetic diseases. But germline editing embryos, raises major ethical concerns involving safety, inequality, and human diversity. This technology requires strong global governance, transparency, and public involvement. Ultimately, CRISPR could shape the future of human health and genetics. The real challenge is ensuring we use it responsibly, ethically, and fairly. Here are my references that I used for this presentation, which would include peer-reviewed scientific sources and international guidelines. These works inform the scientific and ethical analyses in this presentation. Thank you very much for listening to this presentation. I want to thank Professor Watson for an absolutely amazing semester, with cellular biology, and I wish you nothing but great holidays. Have a good one.